Hi guys, we're case study number 16. I'm Isabella and I'm a bio major.
I'm Gabby and I'm a math major.
I'm into an a bio major.
I'm Ann and I'm a bio major.
And our project is on the effects of nitrate on radish growth. And for the abstract, just to give a quick overview of the presentation we'll be giving, basically our goal is to tackle an issue that deals with water scarcity. And so we have an experiment that will be explained by Ann. And our study goal is to determine whether runoff containing nitrate from fertilizer can be used to irrigate radish crops and therefore conserving clean water in developing countries. And so from our results that will later be described by Gabby, we realized that we need further research to make a final conclusion. And then for our introduction, so water scarcity is a huge issue that's global. However, it does affect developing countries in proportional to the rest of the world. And so our project is going to be mainly focused on South Africa and the regions there. And so what we found is that most of the crops that they have are irrigated through precipitation. However, due to climate change, weather patterns have become more erratic and it's become more unpredictable. And to predict the weather and precipitation and raining. Therefore, this has become an issue for crop irrigation. And so when there isn't rain to water the crops, they use clean water, which obviously contributes to a water shortage. And as a result, our project is to see whether we can reuse runoff from fertilizers to see if we can conserve that water in areas like South Africa. And the reason why we chose radishes in particular is because they have a short germination period, which was good for our study since we didn't have that much time. And also it's one of the most common crops in South Africa.
For our materials, we had nitrate fertilizer, 3 gallons of water with different nitrate concentrations, 15 Petri dishes, 5 for each concentration group, 3 groups in total, soil and radish seeds. For our methods, we made 2 different gallons of water nitrate solutions, one 2 milliliters of nitrate per gallon, and the other 10 millimeters of nitrate per gallon, leaving 1 gallon as the control. We then took the soil, watered it. We then placed 6 seeds in each Petri dish, and every day for 11 days, we watered the seeds. At 11 days, we uprooted the seeds, the plants, cleaned them, and weighed them.
So the 2 milliliters per gallon group mimics the typical nitrate concentration in groundwater in South Africa. And now moving on to results. After allowing the radishes to grow for 11 days, we observed that the radishes watered with 2 milliliters per gallon of nitrate solution resulted in the greatest average mass. The radishes watered with no nitrate had the second highest average mass, with the high concentration group resulting in the lowest average plant mass. After calculating the p-value within a ANOVA test, the p-value came out to be about 0.2. Although this means the results aren't statistically significant, there was still a difference in means. The higher p-value could be a result of there not being a large enough difference in nitrate concentration to bring about a statistical significance, or it could be a result of having too small of a sample size with too much variability. If we had the time and resources, we would do this experiment again with more radishes for a longer period of time. This happens a lot of the time in the scientific community, but this just means we would need more time and space to plant the radishes to generate more statistically significant results.
In our experiment, we can see that there was a higher plant mass when water was grown with the low concentrations of nitrate, as there was a difference in mean, as Gabby was discussing. This corresponds to real world situations and in agriculture because in general, there's not a significant amount of nitrate in runoff. Nitrate just plays a crucial role in providing amino acids for protein synthesis. But with high concentration of nitrate, we can see an increased osmotic concentration in the soil solution, which can actually hurt the plants. Reuse of irrigation water can help conserve water. It can help address the water crisis by conserving the water, It can address food insecurity by providing a steady source of food and protect our marine organisms, because runoff eventually go into our bodies of water, which actually hurt our marine life. And also we want to say thank you to Chris Jonah for allowing us to use the CNS research greenhouse. Thank you.